thanks come from me, as I said, to Dr. Passion and to Dr. Yvonne Bryan on behalf of Dr. Passion. Thank you very much for having us, Eric and I, here to speak with you. And we thank Dr. Augustine Diane for all of his leadership over the past few years with our SBIRs. And we feel very fortunate as we bring something out of the university after my 30 years there to the patient. The question is, how do you get your research out of the laboratory into the patient's hands? It's a huge question for many of us. The pharmaceutical companies have it down, but other areas don't. So this SBIR program was sent from heaven as far as I'm concerned. The first slide in my talk about developing biophysically designed beverages is really about my gratefulness to the broader federal government. Funny time to be saying that, isn't it? But you can see there's a ladder of funding in the federal government and even within the NIH for a career. And I was told this when I was in graduate school. You know, JoAnne, you can fund your whole career with this ladder. And as it turns out, when I look back, that's exactly what I've been fortunate enough to do. And I did have a T32. I just saw the name Ruth Kirsten down the hallway. And it should be at the bottom there under 1984. It was my first grant. It was my postdoc. And I've been very lucky, and I tell my students, mentees, and fellows that this is available because many people don't realize it. In 1997, you can see there was a randomized control trial, clinical trial, and I'll be talking about that, about midway through the career. So I'm just pointing that out for you now, that we built a base for a number of years, and the clinical trial was, we thought, ready to go, and then what ensued after that. So the challenge, I tell mentees, students, and colleagues who might need to hear it, but not many do, that it's important to have a career question from my point of view, a big question to answer if you like doing research, and I like doing clinical research. And so that career question, that big question, can be parsed into 5-year pieces. Smaller, but still important questions that can be answered by funding, which usually is 3 to 5 years as we move along. My big career question had to do with oropharyngeal swallowing problems because as I entered the medical world, back as a speech pathologist, nobody was dealing with swallowing problems, specifically oropharyngeal swallowing problems. And so the system, thank you, was being bypassed and G-tubes were being put in the stomachs of patients. And while I was helping patients who couldn't communicate or talk so well, I noticed many of those same people could not swallow well, and unfortunately either were not getting what they needed or getting a G-tube in their stomach, which was unacceptable to me, being somebody who might be someday in that particular situation. So necessity is the mother of invention. Plato told us that a long time ago, and Frank Zappa repeated it in the 60s to remind us, so it still holds true. And it is. Necessity leads to innovation. And that's what happened and is still happening in dysphagia, or swallowing problems. The world, as you know, is facing a situation without precedent. We soon will have more older people than we have children, and more people at extreme old age than ever before. Nothing new to you, but population aging is a powerful demographic force. And this is happening not just here in the US, nationally, but globally. So we need to really address this as we, many of us, approach aging. Some of us are retiring, and that's a sign sometimes. But dysphagia is a population health issue that is age-related in that there are 18 million adults with swallowing problems just in this country. There are millions more children just in this country. Harder to track because they don't have Medicare, they have Medicaid. But you can see that this is a growing concern. Let me now do something a little more entertaining and show you a video fluoroscopic swallow study. Many of you may be familiar with these, but to understand swallowing physiology, anatomy, physiology, and its interaction with what we put in the system, the stimuli, which is food or beverage, we use radiography, particularly X-ray, moving X-rays, fluoroscopy. And we need a radiopaque contrast because the head and neck tissues inside are soft. They're soft tissues. Hard tissue like bone images well, soft tissue like tongue doesn't image well. Barium is the radiopaque contrast that outlines soft tissue structures. So this is a young person swallowing a thin liquid, barium. It's, oh, can we go back? It's about the thickness of water, but it's radiopaque. Notice, and hold it up, how clean and fast that was. You swallow a teaspoon of water in a half a second, less than half a second. So swallowing is very quick, and in that time, we want to see it go down as it did for her into the GI tract, which is posterior through the pharynx into the esophagus. We don't want to see it go anteriorly under the epiglottis into the airway. That's going to be a concern for aspiration, which leads to pneumonia. So if everybody got a good glimpse of how fast and quick normal is, let's move on now to this left side. And this lady had a stroke the day before I met her. Good sound. So you heard the response to the aspiration, which outlined her anterior and posterior trachea. And she did not cough, so she's what we call a silent aspirator, which is a real threat to the professionals in the hospital, to her family, etc. Because she doesn't know, nobody knows she's aspirating, which is why this method is so important. So what am I going to do as the clinician, and I was, we never want somebody to aspirate twice because of us as clinicians, and so we thickened the liquid. Nutritionists have been talking about thickened liquids for many years, and we thickened the barium and gave it to her on her next swallow. Gone, no aspiration. And we repeat that to make sure it's not just luck. So thickened liquid was thought to be a good thing to reduce aspiration and maybe go as far as to reduce pneumonias, but that's a big leap. But thickening liquid for this stroke patient worked for the 24 or 48 hours that she was being rushed off to MRIs, all sorts of tests that are done when you're in the hospital before a speech pathologist or swallowing clinician could really work with her and keep her safe, which is what we do initially for some of the many patients that we see. So the causes and effects of dysphagia are numerous. I want to mention, you saw stroke patients. There are hundreds of neurogenic etiologies for swallowing problems. I'm not going to get into them because of time limitations. Head and neck cancer, many etiologies. The biggest one that's growing is sarcopenia, and we'll talk about that in a little while. But I bet some of you have sarcopenia and sometimes cough on a thin liquid when you don't mean to. to the point that you might even be a little embarrassed at a restaurant, particularly if you're talking and drinking. Okay, I see a few smiles. Maybe that means you do, maybe that means you don't. At any rate, the clinically significant problem is aspiration, but there's also malnutrition, decreased rehab potential, increased length of hospital stays, and as I said, pneumonia, if we don't treat dysphagia adequately and effectively. Of course, then hospitalizations, as you know, cost lots of money, and bounce backs coming back into the hospital within 30 days after discharge have targeted pneumonia. That's probably because you cannot fix dysphagia, which is the cause of so many pneumonias, just with antibiotics. You can treat the pneumonia, but it will come back, and antibiotics again will be given if dysphagia etiology targets aren't the direction of intervention. So we have to really get good at diagnosing and treating dysphagia. All right, so what I'd like to point out here is that bariums that we used for swallowing diagnosis for many years were just taken off the shelf in the radiology suite, and they were designed for upper GIs, lower GIs, certainly nothing about oropharyngeal swallowing. There was a lack of standardization. Maybe even my own hospital. I worked in the VA, which is attached to the UW hospital. I worked in both, and they were using different bariums for their swallowing problems, let alone what Vanderbilt was using where I had been before, on. And we wanted to do a clinical trial. How can you do a clinical trial when you don't even have standardization of your methods? And so the first year of our NIH-funded clinical trial, fondly called Protocol 201 by the NIH, was innovating, making standardized bariums. And we used the nutritionists' labels, largely based in viscosity, of thin barium, nectar thick, and honey thick. But we had data, we had standards. We knew that nectar thick was 300 centipoise. We knew that honey thick was 2,000 centipoise, which is a viscosity measure. And we invented that. We found a partner, a big company, one of the two barium companies at the time, called EZM, now Bracco Diagnostics. And we have become the standard of practice in this country. So I have that patent. The UW has that patent. And it's a very proud thing. And I hope some of you are working knowing that you can do that. We can make contributions we didn't dream of along the way until you get there and it hits you in the face. And you realize it's necessity, the mother of innovation. So this clinical trial, once we realized the first year had to be inventing bariums, lasted 10 years instead of the funded 5. We didn't get a whole lot more money, so it was no cost extensions. Thank you, NIH. I am nonetheless thrilled, and Jerry Logeman was the PI, and I was the person who ran the trial, and there was a big name for me that was fancy, but I don't remember what it was. So as we went along, we realized, of course, if you have the bariums that you're diagnosing swallowing problems and saying a person can swallow this, let's say they aspirate on the nectar barium but not the honey, then what are you going to give them to drink? Because what we wanted to test was, does drinking thickened liquids diminish pneumonia as an outcome? Or does a simple chin tuck diminish pneumonia as an outcome? And I suggest any of you with sarcopenia, tuck your chin if you're choking on thin liquids. It works like a charm. So we really wanted to test that, and we have to then also create the beverages to match the liquids, because if you give someone who aspirates on nectar fluids, they aspirate on nectar barium, they're going to aspirate on their drinks, on their beverages. So you have to match fluids to the diagnostics to eliminate aspiration, which became another part of this trial, but really based on outcomes. We had to learn, well, we needed it for the trial, but we needed something really hardwired based on our outcomes. We had to match our beverages to our bariums afterwards. What we found with another funded study, this was by the USDA, and I think we are the only people, the only lab that was ever funded in swallowing by the USDA. We had learned that thicker, as you saw with our stroke lady, is not always safer for beverages for people with dysphagia. They get pneumonia. Some people with thicker, really thick beverages get pneumonia. We had starch base in our clinical trial, and starch base is hard to cough out, we hypothesize. So we wanted to create beverages, of course, then, that will diminish aspiration, and then had to do a lot of research on what are our best thickeners and what else besides viscosity is important to make swallowing better. And we actually looked at 30 thickeners, with starch and kinds of starch being one group, but we looked at gums and other thickeners as well. And when I say looked at, studied, but I only had 20 minutes here before Eric comes up. So that would take up, talking about 20 thickeners would take up my 20 minutes. So I'm going to keep moving here. We needed to do our rheology research for beverages. This is a rheometer. No, it's not. A rheometer would be thousands, tens of thousands of dollars more expensive. This is a viscometer, a Brookfield viscometer that we borrowed to study the beverages on, in a very discrete way. And we studied, in addition to viscosity, and found that other parameters of rheology, like yield stress, like flow rate, etc., are important parameters for swallowing. May make the bolus move slower, so the muscles have time to kick in, especially in people who have, for instance, a neurogenic issue, etc. But we started to reform a team. I must say the NIH has been kind. I was told by some friends here that my grants would come in and it would be like, oh, damn, there's Robbins again with that huge multidisciplinary group we have to fund. This was 20 years ago. We had radiologists, we had nutritionists, we had speech pathologists, engineers, etc. And it's only gotten bigger. So as this came about, Rich Hartel, one of the finest food scientists who studied chocolate his whole career, and opened up his brain to study dysphagia with me because he had the viscometer on campus that I found, and was kind enough to borrow it, became part of our group along with a sensory scientist named Zeta Vickers at Minnesota. Reach out. I think that's important to tell our students and our postdocs, et cetera. Reach out. There are smart people not right in the walls where you are. At any rate, let's just talk for one more minute about sarcopenia, because my lab in Madison was the first lab to define sarcopenia in the head and neck muscles. So this fella has sarcopenia in his quadriceps. You're seeing the diminished muscle mass, and with that goes diminished strength. And as we age, as you age, as I age, we are becoming sarcopenic, most of us, not all of us, but some of us. It's healthy aging. So we defined it, and we knew that it is prevalent. The NIH had 2 seminars on it, and the Journal of Gerontology put out 2 journals documenting the seminars or the workshop. And we decided we needed protein in our beverages as well, because maybe we could kill 2 birds with 1 stone. And if we have thick beverages to not be aspirated, to build nutrition, we also could put in protein and maybe build muscle mass or affect it in some positive way. Being from Wisconsin, we have lots of cows, so whey protein became the protein of choice. Proteins taste horrible. So this took quite a while recently, and this was the SBIR, what shall we call it, magic, that we are so grateful for, to really get those proteins in, to make acceptable beverages that the patients will adhere to drinking. If you don't have adherence, you have nothing, correct? We all know that. I was thrilled to see that Jerry's were doing palliative care, because one of the things we do is help people choose how to die. Are you going to drink beverages, thin beverages? I mean, it's a huge area and growing. Dr. Miller, I'm sorry. I've known Jerry for so many years. And I think I may have used up way beyond my time and want to turn over to Eric, but I need to say that it's been such a pleasure doing clinical research as a career that I hope some of you are doing it and go home at night feeling lifted by it. Both parts of it come together, and we can make it better. So thank you, the NIH and other agencies and all scientists, and nursing is so important. Education is so important. Not that nursing's the one to do it, but we need to educate about dysphagia, because most people, those who even get it, don't even know how to talk about it. They don't know what it is, they're surprised by it, and education is huge as a need. So thank you so much for letting me share with you all of the wonderfulness of what I've been doing for a while. And Eric has only made it better, being the CEO of Swallow Solutions, helping us get this invention or set of inventions to the patients. Eric Horler.
Okay.
We'll tell you about the solution. No pun intended, solution.
So now I think I have 6 minutes. So we've all climbed the mountain with JoAnne, and all of the science and the research, the clinical work that got to the solution, which you can see on the screen here. Biophysically designed beverages for people with dysphagia that create matched standards for diagnosis and treatment and address sarcopenia. What I'm going to talk about is that unless you can figure out how to get your product into the hands, or in this case, the mouths of your patients, it doesn't matter. And it's not going to make any difference for the population. And so that's where commercialization comes in. There's a lot of different things that go into commercializing any product. So you need to understand your market. You need to figure out how you're going to actually take your product and get it out to the market. How are you going to sell it? How are you going to distribute it? How are you going to manufacture it? You know, are you going to do everything yourself? Are you going to partner? And of course, underlying all of this, can you even make money? Because if you can't make money, again, it's not going to matter how innovative your product is. Being as how I only have 6 minutes, there's no way I'm going to try to address all of that. Rather, what I'm going to do is I'm going to give you an example of just a tiny piece of it as kind of a case study of how commercialization figured into what we did at Swallow Solutions. And the 2 points I want to make, so I want to echo what Augie said about the importance of commercialization. How do you learn about commercialization? You talk to customers. The only thing I'm going to challenge a little bit is this idea that commercialization is phase 3. Commercialization needs to start way before that. Commercialization needs to start at the very beginning, or you need to be thinking about commercialization at the very beginning, or you're going to get to the end of the road and you're going to have something that you can't commercialize. So I'm going to talk a little bit about how we went about gathering the information we needed to make sure that we would have a commercially viable product. Commercialization, just like science, needs to be data-driven and it needs to be rigorous. So we started way back in that phase 1 of that SBIR doing interviews, doing qualitative research. We followed that up with quantitative research so that we could get statistics on what the market was looking for, what the market valued. When it says value prop, that means value proposition. Basically, that just is business speak for what do people care about? What are people willing to buy? And then as we went through the process of doing development, and all of this was in parallel to our SBIR, we were gathering more and more and more data and making course corrections on the product as we went. So just again, using as an example, some of the things that we learned from those first 2 boxes. So the very early research that we did back in parallel to our phase 1. What you have on the right-hand side of this slide is an influencer map. We call an influencer map. So in the center, you've got the person who's the decision maker on the purchase of your product. The size of the circles indicates how much influence any given individual has on the purchase, and the arrows show you how that influence works. So we learned some really interesting things, which are shown in this graph, when we got out and started doing market research. One of the things we learned, notice where the patient is on this map. The patient is really far from the purchase decision. The patient has essentially 0 influence on the purchase decision for this kind of a product. Whether that's right or that's wrong, that's important to know if you're going to try to commercialize it. Also really interesting to us was that the speech pathologist, the qualified clinician who understands the science that JoAnne was just talking about, also has very limited influence on the purchase. That's a challenge. That was something that we had to figure out, how are we going to address that? I mean, if you look at the graph, state regulators have more influence on this purchase decision than the speech pathologist. Dietitians, on the other hand, have a higher level of influence. And then ultimately, with our quantitative research, we were able to go out and test which of the different value propositions, which of the different potential benefits of the product resonate more to different groups. And on the far right-hand side of this slide, you'll see where we have force ranked what the market felt relative to the strength of the influencers were the most important value propositions. And what really floated to the top was protein. So JoAnne said, by putting protein in these beverages, we could kill 2 birds with 1 stone. Commercialization told us, if you don't kill both of those birds with that single stone, then you don't have a stone. We learned this during our phase 1. We were very focused on rheology. We adjusted as we moved into our phase 2 and said, we need to not lose any focus on rheology, but we've got to do it with protein. And one of the things that we discovered was getting protein in a beverage at the correct thickness that tastes okay, and notice sensory, number 3 here, was a tremendous technical challenge. It took us a 1.5 years with some really, really brilliant food scientists to solve that problem. But all of that we learned from talking to the market was what we needed to do. So in the end, this is what we developed. We developed a line of thickened beverages based upon scientific research, safe for individuals with mild to severe dysphagia, matched to the diagnostic standards so that you no longer have the safety risk that came from that mismatch, that were all appropriate as both a primary source of hydration and protein. We learned from the market exactly how much protein they needed to have, and I'm out of time to talk about all of that. All the things we needed to do, and along the way, we actually did manage to generate some new intellectual property. If we had not done early commercialization work and just continued on the path that we started on, we would have ended up with a product that ultimately would have been extremely difficult to sell to the people who were actually going to be purchasing it, and that means the patients never get it. I'd be happy. Actually, I think I did it, JoAnne, in about 7 minutes.
I know you could.
I would, of course, love to talk about all those other commercialization topics. Every single item in that box, if I go back a couple slides, I'm stealing time, Every single item in that box is a separate swallow solution story. You know, distribution, manufacturing, all of that. Every piece of that is really important, but unfortunately we're out of time. So, thank you very much again for inviting both of us. Echo everything JoAnne said. This is really a wonderful honor for us. So thank you, Augie, and thank you to the council.
Are there any pressing questions at this point? You may want to just hit your microphone.
I wonder if have this been used for like patients who've had tubes in feeding tubes for a long time? Is there any potential, like when they're not using their mouths, or no?
I'm not sure I understand, but people who had, do I have to hit this button?
No, it's not.
But people who have had gastrostomy tubes have been weaned from them and used these beverages as they've progressed in their swallowing mastery.
Thank you. Yes, you've answered my question. Thank you.
Yeah, we're proud of that. I got to tell you, I used to say, I'd come home and I'd say, there's nothing like getting $1 million NIH grant. Instead of that, you know what it is? There is nothing like getting somebody off a G-tube, right? That is the truth. Anyway, yeah.
So I hear a lot of, I guess I'm going to challenge that there's nothing like.
Yes.
I hear a lot of sweat equity along the way.
Yeah.
And so I'm just wondering, with your patent, are you making money? Is the university making money? You know, other than the altruistic, oh, I did good. I mean, we're nurses, we do good every day.
Right.
With patients.
And speech
So
pathologists have
I'm just wondering
that.
about the journey and the work and
Yeah.
kind of that bigger pack. How do we
Yeah.
sell this to young people? You know, the sweat equity part and the hours that are put in. And
It's funny because some of my postdocs were just together and they said to me, how did you get the passion? The first patent I mentioned, where the barium solutions came out of our lab, when we're funded by the federal government at the University of Wisconsin, we're a faculty member, the Wisconsin Alumni Research Foundation is kind of a partner of the university, but it's not the university. And you have to bring your patents through WARF, as it's called. And WARF has, if the patent is issued, if we're lucky enough, you know, WARF has the patent attorneys. They work with you very closely. And if we're lucky enough to have a patent issued, WARF has a formula by which the royalties are distributed. So yes, the inventor or the inventors, one of my patents, I had 7 or 8 of my students on with me. And so they got checks when it was being used. I'm sure they didn't know where the checks were coming from because they'd been out of school for a long time. But I think it's fair what was done with the patents and having that formula. My department also, the department is in that formula, and the department gets part of that. So I was the first tenured PhD woman in the medical department, the biggest department in the medical school. And I don't know if they use that patent money in my department or what, but for a long time they gave it to me. Now, this tenured position is part of the faculty going forward. So while I retire, my best mentee will be assistant professor, which is so exciting to me. It's a new program in the medical school, not in communicative disorders or some other school. Did I answer adequately? And if not, I'm happy to talk later.
Maybe one quick follow-up question or finishing question, and then we'll go to the next speaker.
further answer Shirley's question and build on what you've said. It varies by the university. So it's really important if any of us do this that you clearly read the rules of your university, because some universities are more generous than others.
Yes.
So at Penn, when I did it, I started out at 51% owner, and the university was 49%. And then as investors come in, you get diluted.
Are you talking about your patent?
No, about starting a business.
And I was just talking about the patent, so I'm sorry. I
it
misunderstood.
sounds very similar because it is a formula. And so the royalties flow to the department and to the inventor. So we had an invention, so it's a similar thing. But anyway, I learned during that process that other universities will take something like 20% and the rest goes to the inventors. And Penn's was 49%. So it varies by university. And so, that split of when royalties come in, where all the royalties go and they get to you, it really varies. It really varies.
Yeah, and what was nice is I was on the conflict of interest committee of the whole school university. The rotation was for 3 years. They kept me on for 5 years, I think, because I had generated 5 or 6 patents. They wanted to make sure I got it. I understood these rules because they also they change. Yeah. So I'm with you, Shirley. I hope that we all are strong and don't settle for less than counterparts in other professions and of other genders are receiving.
I just wonder if we should be counseling young people when they apply to a university, if they think this is in their future, to be asking about that formula at application. Because the difference between 20% and 50% is quite a bit. And I'm not sure we do that very systematically because we haven't been thinking of that kind of a future in the future.
Right. My last postdoc, who I just mentioned, will be going on as faculty member. The first thing I did when she came into my lab was I made her a WARF ambassador. So an ambassador to the Wisconsin Alumni Research Foundation. So she would learn all of this as she did her funded research. Hand in hand. Thank you.